Okay, so I think this has been a good session, and I'm excited to come after these distinguished speakers. So in the last 20 years, I think the science has started to come together to make it possible to understand growth disorders on a more detailed level. For many years, the large part of the children we see for short stature will get the unhelpful diagnosis of idiopathic short stature. In the endocrine community, maybe we have basically only had one tool to treat growth disorders. And maybe when you're in that situation, you may focus in on just a narrow part of the field, and that's really the goal on my talk here, is to try to widen your perspective on growth disorders. So let's start by just asking a very simple question. So how do children grow taller? So we're all pediatricians, so of course know the answer to that question. That's because of this structure, the growth plate, which is situated at the end of long bones and in the ends of the vertebrae. If you look in a microscope, we can see that it consists of three layers. The resting zone, where stem cells continuously provide new cells to the proliferative and the hypertrophic zone. In the proliferative zone, as the name implies, the cell replicate at a very high rate, and they divide horizontally and then rotate to place themselves along the long axis of the bone, thereby forming these beautiful columns, which enables the formation of bones with an elongated shape. And then further down, they start to increase in size, and this increase in size also makes an important contribution, as I will show you soon, to growth. And this is how it looks. So in the growth plate, you have proliferation, and they're labeled with yellow, which contributes to chondrogenesis. And the cells, as they proliferate, they push the epiphysis away from the metaphysis, thereby extending the bone. And then they also increase in size. And as I said, this makes a very important contribution to growth, as they go from about 7 microns thick to 35 or 40 microns thick in the hypertrophic zone. The hypertrophic chondrocytes are interesting cells because they do not only swell up, they also mineralize their matrix. And this is sort of the way that chondrogenesis is coupled to osteogenesis, and because mineralization of the matrix is required for this cartilage to be replaced by bone. And they do this, among others, by producing alkaline phosphatase, which hydrolyzes pyrophosphate, which is an important inhibitor of mineralization in all tissues. And they also produce factors like VEGF that attracts vessels and bone cells that invade the newly formed cartilage and remodel it into bone. So the net result is that new bone is laid down at the bottom of the growth plate, pushing the epiphysis away from the diaphysis, thereby elongating bones and making children grow taller. But to understand this process fully, we have to understand the growth plate. And you're already aware, and we've talked about this, that there are many endocrine factors that are important for the growth plate. So for instance, the thyroid hormone, that's why children with hypothyroidism are sometimes detected in our short stature clinics, or why children that receive glucocorticoids in excess grow poorly, or why children with growth hormone deficiency or IGF-1 deficiency, as Peter just talked about, do not grow well. There are also many other factors, like cytokines, that can act directly on the growth plate to inhibit growth, and that contributes to the poor growth of children with inflammatory diseases like juvenile arthritis or inflammatory bowel disease. And these factors do not only act in isolation. There are a lot of interactions in between here. So for instance, nutrition feeds into, to a large part, into the endocrine system, and the cytokines also affect the hormonal system. But even more extensive regulation goes on locally in the growth plate. And that is what we are understanding a lot more about during the last 10, 15 years. So there are paracrine factors, and here I'm just listing some of the very important paracrine factors like IGFs, Indian hedgehog, and PTHRP, which forms an important regulatory system to control the length of the proliferative columns. Fibroblast growth factors, C natriuretic peptide, and I'll get back to some of these. And there are even more intracellular factors, so processes within the chondrocytes that, if not functioning optimally, the children will not grow optimally. And then finally, the extracellular matrix. So there's interactions between the extracellular matrix and the cells that are very important to the function of the growth plate. So let me exemplify a couple of these. So let's start with some paracrine factors. So first, the C-type natriuretic peptide. So despite its unfortunate name, its most important physiologic function is in the growth plate. And in the growth plate, it stimulates proliferation in the proliferative zone, hypertrophy, so the increase in size, and matrix synthesis. And that's why it also stimulates longitudinal growth. In the cells, it acts through the MPR2 receptor, the natriuretic peptide receptor 2, to stimulate growth. Consequently, if you have homozygous loss of function, you get a severe skeletal dysplasia called acromesomelic dysplasia Moreau type. And recently it was shown that heterozygous loss of function causes idiopathic short stature, except that now we shouldn't call it idiopathic anymore since it's since we know the cause, and we should call it isolated short stature. And recent studies have shown that up to about 2–3% of children with idiopathic short stature is due to mutations in the MPR2 receptor. Interestingly, if you have, so these are loss of function mutations. If you have gain of function mutations, so this is of course rare, but then you get tall stature, as recently shown by some Dutch pediatric endocrinologists. Okay, so that's MPR2. Another one is fibroblast growth factors. So they are expressed in the growth plate, but maybe even more in the surrounding tissues, and act on the growth plate. In the growth plate, they inhibit proliferation, inhibit hypertrophy, and they inhibit matrix synthesis, and therefore longitudinal bone growth. At the cellular level, they act through the FGFR3 receptor. And consequently, if you have activating mutations, you get, if you have the most severe forms of activation of the receptor, you get thanatophoric dysplasia, lethal skeletal dysplasia. If you have the typical glycine to arginine mutation at amino acid 380, you get the classic achondroplasia syndrome. Whereas if you have slightly milder, you can get hypochondroplasia, which is a disproportionate short stature syndrome that luckily usually do not have the problems of achondroplasia with craniocervical stenosis and spinal stenosis, which is a problem in a lot of those patients. And then finally, you can also get isolated short stature from activating FGFR3 mutations. Okay, so if gain of function mutations give short stature, you say, hey, maybe loss of function mutation causes tall stature. And that is exactly what it does. It causes the Katzels syndrome, and you just have to love that acronym. Very useful, but it stands for camptodactyly, so those are the crooked fingers, tall stature, and hearing loss syndrome. Interestingly, these two paracrine factors interact on the signaling level. So FGFR3 activation overactivation leads to overactivity in the MAP kinase pathway, which is the important pathway to inhibit growth in achondroplasia and other FGFR3 related skeletal dysplasias. CMP, on the contrary, inhibits the MAP kinase pathway, and that's at least part of its effect on growth. And this is, as you may know, now explored. So there's a CMP analog in phase 2 trials for achondroplasia, and there are other biotech companies developing a CMP in the carrier system, and there are FGFR3 antibodies developing. So we may actually see some progress in the possibility to develop to treat some of these syndromes. Okay, so then I said that the extracellular matrix is important. The extracellular matrix in the growth plate is a highly specialized matrix. So it's very rich in collagen type 2 and proteoglycans, where aggrecan is the most abundant proteoglycan in cartilage matrix. If you have homozygous mutations in aggrecan, you get a very severe skeletal dysplasia with adult height. These are the only 3 individuals described, but their height was between 66 and 71 centimeters. Extreme short stature with also severe brachydactyly and severe mid-face hypoplasia. And this was one of only 3 families with aggrecan mutations described. And before this boy came to our clinic for evaluation of short stature. And you can see that he was short, and he was sort of slowly trailing away from the normal percentiles. Interestingly though, in contrast to most children with short stature, which tend to have a delayed bone age, he had a markedly advanced bone age. He also had some other features with mid-face hypoplasia, brachydactyly, and his mother had an interesting growth history. So she says, so as a girl, she was not short, but then at 10 years of age, she just suddenly stopped growing despite having a normal puberty with menarche at 12 years of age. So that is highly unusual to stop growing 2 years before menarche in a girl. Luckily, we had good collaborators, which is always important. So we collaborated with Andrew Dauber, which at the time was at Harvard and medical school, and they had 2 other families, a girl with type 1 diabetes that just suddenly stopped growing, and another girl that sort of similar to our boy was trailing off and had an advanced bone age. And then you can see had a very disappointing growth spurt during puberty. In sequencing, identified heterozygous mutations in aggrecan in all these 3 families, and it co-segregated perfectly. And interestingly, through collaborations, we have now identified more than 20 families with aggregate mutations and been able to more carefully phenotype this syndrome. So you can see they have sort of a mild mid-face hypoplasia, which gives the face somewhat round appearance. In only 3 out of 20 families, but some have these short thumbs with very short distal phalanges. They tend to, so this is the sitting height index, so it tends to be in the upper normal range or frankly elevated, so mildly disproportionate. And as I mentioned, bone age is tend to be accelerated, but this is variable, and some actually did not have bone age acceleration, and now we have found some patients with mildly delayed bone ages. So it is a variable phenotype, but they do tend to lose height percentiles, so they're born close to -1 on average, but again, some are considered SGA at birth, but as adult they have marked to severe short stature. Unfortunately, it doesn't only affect the growth plate, but also other cartilage like the articular cartilage. And I think this is part of why we should do this, because this will help us identify other associated conditions. So in 10 or 20 families reported joint problems, typically with symptoms starting in the second and third decade, with knee pain and stiffness and locking. And if you do an MRI in children, you can sometimes see this osteochondritis dissecans. And in the adults, they develop early onset osteoarthritis. It also affects the intervertebral disc. So in 7 out of 20 families, there was a co-segregation of intervertebral disc disease, again, starting a little later, but still early in the third and fourth decade, with progressive disease, many having neurological symptoms and requiring surgery. So that's an example of a extracellular matrix disorder that can show up as idiopathic short stature in your clinic. But this was, of course, an autosomal dominant form of short stature. Most of the patients we see in our family, though, do not look like that. They look more like this, where a lot of members tend to be of both sides of the family tends to be on the short range, and then suddenly you have a child that's a little bit shorter and come to your clinic. So these are not something that we can work up with genome sequencing at current times, and probably not anytime soon. This is probably polygenic type of short stature. But the genome-wide association studies can actually give some information, because what the genome-wide association studies have done, they have looked at the normal variation of height. And since polygenic short stature is considered to be just the tail end of the normal variation, these studies are informative for polygenic short stature as well. So a large consortium of investigators have done GWAS studies on more than 250,000 individuals and identified more than 500 loci that are linked to variation of adult height in the population. So they were very successful, but what does this tell us? Well, at each locus, there are multiple genes that are sufficiently close to account for the linkage to height, but probably at each loci, only one is really important for growth. So to try to tease that out, we did a bioinformatic analysis where we gathered all the genes in the skeletal dysplasia network and all genes in the MGI, so in the mouse genome phenotype database that has the growth phenotype. And then we combine that with our own gene expression data of genes that are expressed in the growth plate. So we specifically looked at genes that were either highly expressed in the growth plate or regulated either with age or with zone in the growth plate. And then we compared the GWAS list of genes to this growth plate list of genes and found that it was a highly significant overlap. And what we found was many of our friends here as endocrinologists, like the growth hormone receptor, IGF-1 receptor, aromatase, yeah, you recognize some of them. We found paracrine factors, again, the same ones that I've talked some about, and some additional. We found genes related to cell cycle regulation, which makes sense since there's a lot of proliferation going on. And then many matrix-related genes, including our friend Paracrine here. So I think that just points to the same large network of genes that we can find in skeletal dysplasias are also important to regulate normal variation of height and therefore also probably what's in between the polygenic short stature. This is another list of genes that recently been reported in the idiopathic short stature group of patients. And here you can see that we're now with combined efforts starting to find tease out the idiopathic short stature group. And if you put them all together, there's probably about 10 to 20% of the idiopathic short stature group can be explained by monogenic isolated growth disorder syndrome. So like SHOX has been reported very variable depending on what cohort you use, from 1 to 22%, but it's probably the most common cause of idiopathic short stature. MPR2, as I mentioned, about maybe 2%. Agarcan, 1 to 2%. Indian hedgehog, this is just one study maybe, and that's from the same group that from Alex Horsch's group in Sao Paulo. So they may have more of those than others have seen, but still a significant amount of patients. So I think we are moving towards that it makes it rational to soon consider maybe sequencing a panel of genes in children with severe isolated short stature. So with that, I'd like to conclude, and I hope I convinced you that the growth plate is the center of attention when we talk about growth, and that growth plate chondrogenesis is the fundamental biological mechanism that drives growth, and therefore all short stature is due to decreased growth plate chondrogenesis, and essentially all tall stature to increase growth plate chondrogenesis. Consequently, many regulatory systems important for growth needs to be considered when we're talking about short stature and evaluation of short stature. And finally, something that we maybe didn't talk about, but you can realize from my presentations is that there's a wide, in each growth gene, there is a wide phenotypic spectrum depending on the type of mutation and inheritance pattern that can go from severe skeletal dysplasias to short stature to normal variation of height. And in some cases, even the opposite phenotype with tall stature. With that, I would like to conclude my talk. Thank you.